Well, it's basically you.
Can anybody hear me? Can anybody hear me on the computer? Yes, Lupe, can you hear me?
Yeah, I can.
Okay, all right. Once again, if you can, put your name in the chat so I have a record of you being here. And we'll stall a little bit till other people come. Okay. We're almost at 9 o'clock. Good morning.
Morning.
Okay. All right. Good job, people putting their name in the chat. Thank you. Okay. First of all, I want to remind people that we have a lab exam on Friday. And so on Friday, be there at the regular time. The door will be closed, but I'll open it right as the time turns, and we'll go through the exam. It's going to cover the lab exercises from the first through the end of the skeletal system. About a third of it will be skeletal system components, and the rest, there's quite a bit of skin and tissues, and then some microscopes and some terminology type stuff. I also wanted to look at the schedule for the working with the textbook, so let's look at that, see if I can find that. Okay. So this is the lecture schedule and assigned readings. Does anybody see that? Yes, thank you. Okay, so we're in the week of October 5th, and we're working on chapter 10, the muscle tissue and types of muscles. And next week, I'm going to do chapter 11, and the second lecture exam comes next week, which will probably open up on Friday again, and it'll be open Friday and Saturday. It will be about the same length as the previous exam, and it will cover chapters 5, 6, 9, and 10. Okay, so we're kind of finishing up some of the new material today and this week. I'll have some review stuff for us for next week. We'll go over some muscle ID next week and talk about how structure of individual muscles. There'll be a little bit that might be on the exam, but much of what we'll do next week will actually be tested on the second lab exam. Okay, so we have lab exam this week, and then next week we have a lecture exam. But this is midterm. We're right in midterm, so this is kind of when you have to expect it. Okay, does anybody have any questions?
What time is the exam?
For the lab exam?
Yeah.
The lab exam, for some people it will start at 10 after 10, and for the others it will start at 11:15, depending which lab group you're in.
Okay.
Okay, so let's, what should we do here?
A schedule, or
Pardon?
with what you say, some people will take it 10 to 10 and some take it 11:15. I mean, I want to be sure to be there in time because they drive far away.
Yeah. So Anna, I think you're in the second lab group, which is normally at 11:15, and that's when you'll be there. Okay.
All right, thanks.
Okay.
Can I ask one question too?
Sure.
How important is spelling on the lab exam? Because some of those words are crazy.
Yeah.
Is it like if I put an E instead of an I, am I still going to get it right? Or
yeah, yes. Now, hold on here. Let me share something else here. Oh gosh, I got to figure out how to do this. I haven't used the whiteboard yet. This is just a chance to use the whiteboard. So they're spelling. If you spell, let's see if I can do this. I can't do this. Can I do this? That's not going to work. So, forget this. So, spelling, spelling if it sounds right, that's good. And so if you miss an I and an E, that's okay. If you flip them in order. But there are some words that, like if you get halfway to one and halfway to another. So the lower, or the two bones of the lower leg are the tibia and the fibula. If you write fibia, that's gonna, even though it's only one letter, it's half of one word and half of another. And so I've got to, I can't take that. If you write tibula instead of tibia or fibula, I'm stuck. And so as long as I know, if I can figure out where you're heading, but if you're halfway between one and halfway to another, then I'm stuck. And so try and get there. And so errors, if you shift one letter and it's 10 letters, that's not a big deal as long as it doesn't seem like it's something else. And so I think you know what I mean, Heather. So okay, do your best. Usually that's good enough. Okay. All right. Let's see. Okay. Let's look at some of the PowerPoint slides. Okay. We'll go from the first one. Okay. So there's a lot of new stuff this week. A lot of this is it's just really new and it's different. It's nothing anyone's ever heard. So a lot of, we want to talk about, hold on here. I gotta see if I can get this. Muscles, tissues, and we're talking about the muscular system, which there's functions of the whole system like voluntary movement, walking, talking, moving air in and out of our lungs. Those are really important. Posture, holding us erect and in position. Communication, movement in organs, these are all functions. But we have to go into the specialized cells and tissues of muscles. And so this gets to be really complicated, and the mechanism of contraction. So muscle cells are cells that have a super souped up cytoskeleton. And so their cytoskeleton takes up virtually all of their cell. Okay, and we talk about the proteins actin and myosin. These are proteins that are just slightly modified cytoskeleton proteins. And cells can normally change shape and move, but muscle cells can do it to an extreme. So they're filled with these myofibrils, which are bundles of proteins regularly arranged. Okay, and so that's kind of the main feature of these cells, is these bundles of proteins. And then they have other stuff that are needed to get the proteins what they want. So they have a modified endoplasmic reticulum to store calcium. They have a modified cell membrane so that they can receive messages from the nerve cell, and that they can spread an impulse into these tubules so it gets to the sarcoplasmic reticulum. Then generally these cells have lots of mitochondria, and they're so elongate that they have more than one nucleus. Okay, they're just unusual cells. It's really the myofibrils that make them unique. And so I give you this other picture. They're basically filled with these proteins that are involved in contraction. And these proteins are organized in repeating units. So the myofibrils have the thick filament and the thin filament, and those thick and thin filaments in skeletal muscle and cardiac muscle are really regularly arranged in these sarcomeres, and so you can see these stripes under the microscope. Okay. Now we go into the molecular details of how these things work. The filaments shown here, that little picture that has actin and myosin, Okay, the thin filament is actin, and actin has two other proteins wounding around its surface, tropomyosin and troponin. And normally that tropomyosin prevents actin and myosin from interacting. It's only when calcium comes and binds to troponin can then myosin grab onto actin and the filaments slide. Okay, and so these filaments, lots of this kind of sequence of events here is to encourage the release of calcium ions, and then how we get to the release of calcium ions, how calcium changes actin, and then actin and myosin interact. Okay, and so you know, that's going to be probably the hardest thing in this chapter, is this kind of figuring out this interaction. Muscle cells have lots of actin and myosin. To contract, they need to release calcium, and they need to have ATP available. And then what happens is the myosin grabs on and just pulls the filaments past one another. Okay. All right. So let's go to, I have a little more detail of that in the second program, which was the physiology one. So let's go look at that. You see the neuromuscular junction slide? Yes, thank you. Okay. So the top thing that comes down, that's the, this, I don't know if you can see my thing, this top part, that's the end of a neuron. And this bottom kind of bluish line here, that's the membrane of the muscle cell, or the sarcolemma. So to stimulate a muscle to contract, a nerve cell releases a chemical called a neurotransmitter, and for all of our nerve cells, this is called acetylcholine, and it's abbreviated ACH. Okay, and ACH is going to be released by the vesicles of the nerve cell, and then it diffuses across this short gap, which is a very, very short gap, like tiny, tiny, tiny. So it can cross that very fast, and it binds to a receptor on the muscle cell membrane, and that receptor is also a channel. And so this is the closed channel, the ligand-gated sodium channel. When acetylcholine binds, it opens, and that allows sodium to enter into the cell. Since sodium carries a charge, that causes the cell to change its charge, and this action potential spreads across the surface of the cell. Okay, as long as acetylcholine is active, these things will be open. There is an enzyme that gets rid of acetylcholine, so our muscle cells aren't overstimulated. So this is the first step, the neuromuscular junction. The second step is called excitation coupling. It's where that action potential gets out and it travels across the muscle cell membrane and then down the T-tubule. Okay, when that change to the T-tubule, that causes the T-tubule to release calcium ions. So the excitation that occurred here is actually going to excite the actin and myosin. So this is why it's excitation coupling. Okay, and then that leads to the sliding filaments, where the calcium had shifted the head of, or shifted troponin, and then the troponin shifts tropomyosin on the actin. So the actin has now exposed binding sites. And so myosin has these heads that grab onto these binding sites. And when the head binds to that, it changes its position to slide the filaments. And so you get this little arrow sliding the filament. This is under 2. Okay, the filaments will remain together. Okay, unless there's ATP. If there's ATP, the head will release the actin, and it will resume to the cocked position, And as long as calcium is still available, it'll grab the actin further down. And so these heads are just going back and forth, back and forth, back and forth, as long as there's ATP available. Okay. And so this will continue as long as there's ATP and as long as calcium's released. To stop the muscle contraction, the sarcoplasmic reticulum withdraws the calcium. Once it withdraws the calcium, when the myosin head releases, then it can't grab onto the actin again. Okay, if you run out of ATP, okay, then the myosin head is contracted to the actin, okay, and it can't let go. This is what happens during a cramp. Okay, in a cramp, like an athlete, they can have this. They run out of ATP. You can get these at night because blood flow to particularly your legs really decreases at night because your heart rate drops maybe down into the 50s or 40s at night. And so you get less oxygen going to the muscle. And after a couple of hours of this, you may have used most of the ATP and you wind up with, less ATP down there. Okay. And then the last thing I talked about in this physiology program was the energy for muscle contraction. So muscles use a tremendous amount of ATP, tremendous amount, and they can use it really fast. There's two general sources. One is aerobic. In aerobic sources, that means with oxygen. These cells they're using oxygen to break down glucose or fat. They do that in the mitochondria. And because it requires oxygen, these muscles have lots of blood vessels, and they actually store some oxygen with a pigment called myoglobin. Fibers that do this best are called slow oxidative fibers, and they appear very dark red because they have this really good blood supply and they have myoglobin. We find these in our postural muscles, in our leg muscles, things like that. Okay, and these fibers are resistant. They're not going to ever muscle fatigue. Okay, they can make oxygen as fast as they use it, or they can make ATP as fast as they use it, as long as the oxygen supply is good. Now, the other type of energy source is anaerobic. Okay, this means without oxygen. Okay, and actually, anaerobic reactions in muscle cells make ATP faster than aerobic reactions. However, they use up glucose at incredible rates, and they can only use glucose. So these muscles have to have lots of glucose, and they use it up very, very quickly. And they also have really fast myosin. So these muscles will contract much faster, okay? But they use up glucose, and the waste product they produce is lactic acid. That changes the pH of the muscle. These muscles fatigue. Okay, and so these are like sprint muscles. You generally have a lot of these muscles in your chest and arms. They're good for like really powerful stuff. Training can make your leg muscles more like this. These are called fast glycolytic fibers. They also they don't really have much blood supply. They don't store myoglobin. They store glucose, but they fatigue quickly, really quickly. 10 seconds, 15 seconds, something like that. I did track in high school, and it always used to really bother me. I ran the 1 mile race and then a 2 mile race, and that's aerobic metabolism. And so those would be 4 minutes, 9 minutes of just running pretty hard, and you'd run about 15 miles an hour for those kind of races, which isn't that fast. Okay, it's hard to do that for 10 minutes, but it's not that fast. The sprinters are actually running close to 25, 30 miles an hour. Okay, it used to really bother me because I thought, oh, I'm working really hard here and I'm doing it for 8 minutes. And my friend, this Mark guy, he ran 60 meters and 100 meters races. His one race was 6 seconds. It was 6 seconds. I'm thinking, that's not fair. I'm running 10 minutes and he's running 6 seconds. But what I didn't realize was that he had to use an entirely different system, and he was generating all this lactic acid in 6 seconds. Whereas I was using aerobic metabolism and I wasn't really generating hardly any lactic acid. It was actually easier for me to run a second race than for him, because if he was doing his best, he was going to generate so much lactic acid that his muscles were going to be fatigued probably for at least an hour. So it was something I didn't understand, but now I do. It's completely different things. There'll never be somebody that runs a mile at the same speed that these guys can run 100 meters, because they're using completely different systems, and you just can't do it. So, okay. Does anybody have any questions? Yes, maybe?
Do you mean just general questions or?
General questions, yeah. This is the hardest spot. The sliding filaments and the muscle stuff, the energy sources. This is the hardest stuff of this chapter. At least I think so.
I'm just trying to ask some questions about the exam, actually.
Okay.
Yeah.
The
so I wanted.
the lab
sorry.
exam?
Yes. Yes.
Okay.
I wanted to know if some of the practice review questions would actually come out on the exam itself.
Probably, yes. But I'm
Okay.
not going to say they're all going to be there, but there'll probably be some of the same questions. Yes.
Okay. And okay. Sorry.
Go ahead. Well, go ahead.
Okay, I wanted to know what parts of the bones should we really focus on, because there are lots of them,
Well, I think the first thing is to know the name of the whole bone. Okay, I will
Okay.
ask more often. I'll say name this bone. Give me the name of the tibia, the femur, the frontal bone, the mandible, the occipital bone. I'll more often ask that than I'll say name this hole or name this bump or tell me what this structure is. Usually it's name the bone. Okay.
Okay.
My feeling is that most of us don't know those names, and that's the first task. Okay. The second task is know the landmarks, and those tasks can help you. One can help you learn the other. But if you haven't done the name of the bones, I'm pretty sure you don't know the names of the landmarks. So I don't need to ask as many of those.
Okay. Okay.
Girl, I'm
Okay.
stressed.
Lucretia, did you have a question?
I just feel like I'm not going to pass this exam.
Well, work at it. It's repetition. I would definitely write all the names of the bones out. That's something I would do. Writing helps. That's what I'm going to ask you to do. I'm going to ask you to write them out. Okay. That, you're going to have to, from memory, look at this and then write the name of the bone. So one of the study methods I would certainly use is writing the names out. I would definitely do that, because then you've written every name out before. You'll know what it feels like to do that. Flashcards are helpful. Look at some pictures. But and different methods will work for different people, but repetition works for everybody. So find something that you like and do it several times.
Okay.
You still have a couple days. Certainly I would write the names of all the bones out at once a day at least. So I've done that. Okay. I'd have the picture in front and I'd just write them out. That'll help you. Okay.
Okay, I have a question. The bones that you gave us on that list, is those the ones that we are supposed to know? Because our lab it gets pretty precise in our lab books and in our.
Yes. I gave you the sheet because I think those are the most important ones.
Okay.
The lab manual has more detail, especially with the landmarks.
Yes.
And if you go work if you get a job in a clinic that's an osteopath, a guy that does surgery on shoulders, or, you'll have to know every bump on the scapula and every bump on there. I'll trust you'll do that. But no matter where you go, you need to know the name of the humerus, the scapula, the clavicle. Okay. And so that's where we're going for now.
Okay.
Okay. Those are
What
good
if we
questions.
On the right side and the left side?
Yeah. I'm not going to ask you on every bone, but potentially I could ask you. So, and often if you look at the bone and kind of, if you look at them, you can kind of tell there's a right and left to them. Particularly when you get the bones in your hands, it's something you can figure out.
Okay.
So if you take the bone in your hand and you think, is this a right or a left? Kind of say, okay, I think this is a tibia. I can reach down and feel my tibia, and I feel this crest. So this crest has to be in front. And then I can think, is it this way or this way? And usually you'll know. Often, like, the femur has a curve to it, and the lateral and medial condyles, which form part of the knee. If you put the femur on the right side and it's supposed to be a left, it'll look ridiculous, because you'll say, what are these two bumps projecting forward? That's not what it is. But if you turn it around and put it on the left side, it'll look like, yeah, it could be there. So you kind of can figure that out as you're going.
Okay.
Okay. It's not as hard as you might think.
Would we get credit if we actually maybe kind of mix the answers? Like maybe this was supposed to be the left and this was supposed to be the right, and we just like did the opposite. Would we actually get credit for that?
Well, I don't. I don't want to say yes, because I don't. You're aiming for more than getting close. You want to get the right answer. But there are certain mistakes that I'll say, oh yeah, okay, I'll give you half credit. Okay. Those are all good questions.
How much is this lab is worth?
This is like one lecture exam. So same as one lecture exam. So we have.
So is it 5 points or 100?
This is worth 100.
Oh, wiki.
It's okay. Don't panic. Remember the math anxiety stuff. That all applies here. Keep calm. Take deep breaths. Study. That's how you calm yourself down, is by studying. Write this out. If you've written these bones out 3 or 4 times and looked at the thing, you won't be as nervous. Okay.
I have a question.
Yeah.
Mr. Nawaki. Is there any way we can prolong this Zoom lecture section, because technically it should be longer, right?
No, can we prolong this? I can meet with you on Zoom, but we're not replacing lecture minutes minute per minute. Okay. I give you very, very compact lectures that you can look at any time on Moodle, and those are meant for you to look at, study them, pause them when you need to, replay sections where you might. I'm covering the same material, but not in the same time. I'm giving you extra resources. Okay.
Yeah. So, in addition to the book itself, I guess the challenge there is the back-to-back exam. Like Friday's going to be exam, and then having to hear that there's another exam. Now, I'm not quite equipped in a way of having to think to not stress. I'm starting to stress out. It's not a good feeling.
No, I know. Well, they're not, when you say back-to-back, that means they're in adjacent weeks. So there's a whole week separate.
Yeah, sure.
And that's the same schedule we would have regardless of whether it was online or not online.
Yeah, but the retaining of the education that I am getting is not quite, I'd say, it's not even in the average. I'm not getting anything. I mean, I'm getting something through reading and listening to the slides. It's just, I just feel bad about it because I want to take the education that I am getting from this class somewhere.
Okay.
I don't know. I'm just worried.
It's okay to have a little worry about it. Repetition is what matters. If you have questions, you can email me and I'll try and get back within a day or so. And if you say, Ms. Nowicki, I have more than one or two questions, can we set up a private Zoom meeting? I can do that. But I will, that's got to be limited with specific questions. And we do have tutors, okay, and study groups available through the Success, I think it's the Success Center. And those, I think there's one or two available that are online, and they'll set up times. It's, normally what I try to do is have people study in groups because it helps to work together in groups. And obviously this time we can't, but we can set up a Zoom group too as well, if there are several people that want to work together. But there's no way I can just give it to you. You've got to be able to practice it. And so that's repetition, that's writing it out. That's reading and taking notes, things like that. And it does go fast. That's part of the issue, is that we go fast. We have to cover all these organ systems in a semester. And next semester, there's a whole new set of organ systems. That's the nature of it. And the nursing program will go even faster, because you'll talk about, here's how we assess this. Let's go deal with patients. It's you're not going to feel comfortable until you're comfortable feeling uncomfortable.
Oh, I understand that part. I just thought that, signing up for 3 days, having to really think, I guess I'm still adjusting the
Right.
normal in a way, because, I sign up for Monday, Wednesday, and Thursday, but we meet half an hour on Wednesday and some on Friday, and then I get to do what I need to do the rest of the week.
Right.
It's
Okay. All right. Well, why don't we send a couple emails back and forth, and maybe we can talk about some options for you. Okay.
sound good.
Okay. All right. I think we're over a half hour. It's going to cut me off in about 5 minutes. Does anyone else have questions? Okay. I'll talk a little bit about muscle responses in our meeting next week, because I'll have time to do that. And I'll also, the review materials will be ready for the lecture exam probably next Monday. And so I'll give you some idea of those. And those will be similar to what we had last time. So there'll be like a practice test, some review slides that I'll go through in the meeting next time. Okay. All right. So I'll post this online. Again, if you haven't texted or put your name in the chat, do so now, so that we can do. Okay. I hope to see you Friday. Come at the regular time. All you'll need is a pencil. I will give you an answer sheet, and we will be done at the regular time. It's shorter than a typical lab exam, so it's fewer questions, about the length of the practice exam we had the previous Friday. So, okay. All right. Thank you. And I will see you on Friday.